Namaste to all students. Myself, JV and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about cell junctions. And this session is related to cell biology course of program BTech FBT and BSc FST second sem. So what are the cell junctions? So cell junctions serving all of these functions can be found in the cell of different types, and here we will look at each of them in turn. And cell junctions are basically responsible for the connection in between neighboring cells so that they can interconnect with each other. They can, if they want to require some exchange of their material, plasma material, cytoplasm material, or any kind of a secretory material, so they can use these junctions for the transportation process and for the connection process. So there are various types of cell junctions in which plasmodesmata is very famous, and it is present in the plant cells. So plant cells, surrounded as they are by cell walls, do not contact one another through wide stretches of the plasma membrane the way animal cell can, because plasma cells are having extra layer on it which is known as cell wall. However, they do specialized junctions called plasmodesmata. Singular term is plasmodesma. Places where a hole is punches into the cell wall to allow direct cytoplasmic exchange between two cells. So this is known as, this type of a junction is known as plasmodesmata. Here I am sharing the screen of diagram by which you can understand more about the plasmodesmata structure. Here you can simply see this is one plant cell and this is one the second plant cell, and plant cells are having cell wall. So they are not interconnected with each other very broadly, but at one point there is a punch in between two cells. So their cytoplasmic material can exchange directly from this punch area, and this punch area is known as plasmodesma. Plasmodesmata are lined with the plasma membrane that is continuous to with the membrane of the two cells. Each plasmodesma has a thread of cytoplasm extending through it, containing an even thinner thread of endoplasmic reticulum. Molecules below a certain size move freely through the plasmodesmatal channel by passive diffusion, and the size exclusion limits varies among plants and even among cell types within a plant. Means there is no kind of a energy consumption during the passage of material. They are simply based on the passive diffusion, means they will molecules will move from their higher concentration to their lower concentration, means they will move according to their concentration gradient. And the plasmodesmata may selectively dilute to allow the passage of certain large molecules such as proteins, although this process is poorly understood. Next type of a junction, cell junction, is a gap junction. So functionally, gap junctions are present in the animal cell. They are lot like a plasma desmata in the plant cell. They are channels between the neighboring cells that allow for the transport of the iron, water, and other substances. Structurally, however, gap junction and the plasmodesmata are quite different. In vertebrates, gap junctions develop when a set of six membrane protein called connexin form a elongated donut-like structure called connexon. Means six membrane proteins of connexins will create a donut-like structure which is known as connexon. And when the pore of donut holes of connexon in adjacent animal cell align, a channel forms between the cells. Invertebrates also form gap junction in the same way, but use a different set of protein called inaxin. So, for the vertebrates, it will be connexin, and for the invertebrates it will be in axons. Here you can understand this gap junction concept, structural concept by this diagram. These are the two plasma membranes of this one is for cell 1, this one is for cell 2, and this single molecule is known as connexin, and there are six membranes are arranged like this. They will create a donut like structure which is known as connexon, and when it is closed so it will be look like that, and if it is open so a central pore will be opened in between this structure. And this one is a transfer section. You can understand more this channel or this gap junction, how it works and how it look like. Now come to the next type of junction, cell junction, which is tight junction. So not all junctions between cells produces cytoplasmic connections. Instead, tight junctions create a watertight seal between two adjacent animal cells. Now come to the tight junctions, and not all the junctions between cell produce cytoplasmic connections. Instead, tight junction create a watertight seal between the two adjacent animals. At the site of the tight junction, cells are held tightly against each other by many individual groups of tight junctions protein called claudins, each of which interacts with a partner group on the opposite cell membrane. The group are arranged into strands that form a branching network with larger number of strands making for a tight seal. You can understand this tight junction by this diagrammatic representation, in which you can simply see there are so many strands which create the network. This one is the cell A, this is cell B, and this is one single claudin which is interacting with the second claudin, and they are making a tight junctions, and that's how these are the groups of the tight junction proteins. They create a proper network. For the purpose of tight junction is to keep liquid from escaping between cells, allowing a layer of the cells, for instance those lining an organ, to act as an impermeable barrier. For example, the tight junctions between the epithelial cell lining your bladder prevent urine from leaking out into the extracellular space. Next type of a cell junction is desmosome. So animal cells may also contain junctions called desmosomes, which act like a spot weld between the adjacent epithelial cell. A desmosome involve a complex of the protein. Some of these protein extend across the membrane, while others anchor the junction within the cell. Catherines, specialized adhesion proteins, are found on the membrane of the boss cell and interact in the space between them, holding the membrane together. Inside the cell, the cadherine attached to a structure called the cytoplasmic plaque. Basically, read in the image at the right side, you can simply see in the next slide, which connect to the intermediate filament and help anchor the junction. Desmosomes pin adjacent cells together, ensuring that cells in organ and tissue that stretch, such as skin and cardiac muscle, remain connected in an unbroken sheet. You can understand simply by this diagram, where you can see there are two plasma membrane of two neighboring cells, and these are the attachment plaque, and this one are the cadherines which are attached to the intermediate filaments. So this is the sum up of this session, and if you are understanding properly this session, so you are able to give answer in context of the competitive exams. So questions can be asked like what is cell junction, and plasmodesmata will be present in plant cell or animal cell, and tight junctions are there, and conaxin protein is involved in which kind of a junction, and in axin uh, protein is also present in which kind of a cell junction. So you have to give answers about these all junctions, and their concept and their structural differences in between basically. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.